Okay, hi everyone. Happy Friday. Um, Before we start into the lecture today, one of your classmates wanted to share with you some information about a fundraiser that a number of the on-campus Manoa clubs are supporting. And this is to help raise money for the Hawaii Home Project. So Emily is representing one of these clubs, and she asked me if she could have a couple minutes to let all of you know what this fundraiser is. I think it's a very worthy cause, so I told Emily that it'd be fine if she took a couple minutes just to introduce this before we start in with our day's lecture. So Emily, if you want to unmute yourself, you can turn on your camera and share with the class what this is about.
Hi, Dr. Shen. Thank you. Hi, guys. So this fundraiser is, as Dr. Shen said, a joint fundraiser for a lot of the clubs on campus. And I'm actually the vice president of Chemistry Club. And so that's the club I'm affiliated with. The fundraiser is going to be benefiting the Home Project, which helps with the homeless population on island. They do mobile clinics, and they're kind of helping with the COVID situation right now with that population as well. And Malama Mentors is for children in like middle school and high school that are withdrawn, and they're paired up with a mentor to kind of help guide them. And it's another beneficial program. All the proceeds for this fundraiser are going to be split 50-50 to go to those two organizations, and we're collaborating with HOSA, PMA, Pre-PA Club, Golden Key, and actually the NSSA, the Natural Sciences Ambassadors. So I'm sure there are a few of you in here today as well, like our classmates. And so all I can say is please sell as much as you can. And then for those of you who are not, oh, thank you, Ange, for the link to the order form. And the item itself is actually a medium-sized boba milk tea, and you can have three choices, either oolong, black, or jasmine tea, and they're all e-tickets. So once the pre-sale date is over, we'll be emailing you the e-tickets. You can go to Cloud9 Cafe to redeem the tickets, and I really hope that you guys will kind of have fun selling and also just support our fundraiser.
Okay, good, Emily. So, in the PDF version of today's lecture slide, hopefully this slide comes up a little bit clearer. But if they want to have additional information on this, can they reach out to you, Emily?
Yes, no problem. I just texted into the DM, so I have my email and text message as well. And Ange also put the link to the order form. So if you're just an individual buyer and you would like to fill out the form yourself, you can do that, or you can contact a seller from any of the clubs that are participating and have them fill out a form for you as well.
Okay, good. Thank you. So again, I think the Hawaii Home Project and the Malama Mentors definitely very important and worthwhile causes. And so it's nice to be able to kind of help out and get a nice boba drink.
Yep, and you're supporting a local business.
And supporting local business, right? Okay,
Wins
thank you,
everywhere.
Emily.
Yep, thank you.
Okay,
Thank
great.
you, Dr. Shen.
Okay. And thank you to all of you in the class for listening in on that. All right, so let's go ahead and continue our lesson for today. So as a reminder, on Monday, we have the third lecture that's going to be talking about T cells. And so that will be still some new material on Monday. And then next Wednesday will be our review for the first exam. And then on Friday of next week, that is February 12th, will be our first exam. I'll talk more about the exam format on Wednesday when we review. But just so you know, the exam is multiple choice. It's done entirely on Laulima. The whole class takes it at the same time during our normal class time, so from 12:30 to 1:30. So you just want to make sure that you're in a good place, whether at home or library, whatever, where you have good, reliable internet access and your connection's not going to somehow drop, right? Because that'll cause you a lot of stress and anxiety. But anyhow, I'll talk more about that. But I just wanted to remind all of you, because I know many of you probably haven't looked at the semester calendar for a while. Next week, Friday, is our first exam. All right, so this weekend, start doing some studying. All right, so in this slide here, this helps us kind of think about how our innate side of our immune system and how our adaptive side of our immune system are, on the one hand, different sides, because the cells, proteins, things like that that are part of each are handling a different kind of concern or different function, right? I mean, both sides are meant to protect us against things like pathogens, but hopefully by now you have a good sense that the innate side, so the left side of this image here, and all of the cells that are sort of part of our innate side of our innate system, right? That's the more primitive side. That's the side that dates back, not just a million years or 100 million years, but way back to even simpler life forms that have to have some sort of innate way of defending themselves against things like pathogens, right? So that's why we always, for this class, we kind of, in a fun way, hopefully it's kind of fun, we think of our innate side as being more concerned with dealing with typical pathogens that we encounter every day of our lives, right? Whereas our adaptive side is the side that evolved later on, right? So our adaptive side, in relative terms, is only a few 100 million years old, right? So it dates back to when vertebrates first evolved. So early fishes, for example, early bony fishes. And as life forms began to live longer and they were more complex and larger, right? The threat of having some sort of new pathogen come along and having to face that became a greater sort of problem to worry about, right? For example, if you're a tiny little fly, right, and as an adult fly, you live for like 5 days or something like that, then there's no real evolutionary motivation for you to come up with some elaborate adaptive immune response, because in those 5 days, you live your whole adult life, and then that's the end of you, right? So you can get away with just having a decent innate immune system, right? And even though, yeah, you might say, yeah, but there are some flies who are going to encounter something really bad that's new, and well, so what? They're going to die. But then you have all the other flies who actually make it through their 5 days, without encountering something super bad, right? And so evolutionarily, there wasn't much motivation for developing something fancier or more elaborate, right? But once you get into complex organisms that live not for 5 days, but maybe, we're talking about 5 years now, or in our case, 70 plus years, then it's guaranteed that in the course of our life, those many years, we're going to run across exotic pathogens that our innate system can't handle, right? So that's why we needed to evolve, or life needed to evolve, an adaptive immune system to come up with some way of, like we've talked about before, identifying that something is a threat, coming up with a way of being able to pass that identification on to other participants in our immune system, other cells, and then to be able to mobilize a very strong and powerful defense against it. Because by the time your adaptive immune system gets around to kind of waking up and being fully mobilized, enough time has passed that something like an exotic virus, bad clown here, has had time to really start doing a lot of damage and is kind of out of control, right? So you have to have a super strong defense. And importantly, what? If this made you super sick and you're on the verge of dying, but your adaptive system actually did wake up and successfully fought off bad clown, right? Bad clown is in your environment, right? That's how he got into you in the first place. Nothing's to stop bad clown from getting into your system again. You don't want to always start from square one, right? Every time you breathe in bad clown. So having a memory of bad clown, your adaptive system being able to remember and learn, that's why we call it adaptive, right, is vital for us to be able to, in the future, take care of fighting off bad clown before bad clown has a chance to make us sick again, right? I think these days, of course, everybody has vaccines on their mind and vaccinations against COVID, right? And so that's very much the principle here of why vaccines work, right? To provide the memory of a particular pathogen so that your immune system has the chance to prepare to fend it off when in fact you do encounter it, without having it first have the terrible consequence of potentially killing you right, as your adaptive immune system struggles to learn. All right. So notice that certain cells do overlap. So there are certain types of T cells that do sort of function both as adaptive or innate. Natural killer cells, we already looked at an example of how the natural killer cells kind of evolved as sort of the first kind of adaptive side of our cell lineages of the stem cells that give rise to these different types of cells in our bone marrow. But in reality, the natural killer cell still functions more like an innate effector. On the bottom of the slide is a important distinction here that we are going to expand upon today. And so it's important that you get this. Right? Typically, innate effector cells act directly on the pathogen. So things like macrophages, the very first innate effector cell that we learned about this semester, right? It goes around in your tissues, crawling around, and it just eats things up to destroy them. Even neutrophils, they get recruited if the macrophages can't handle it, and then macrophages call for backup, right? As part of your innate system, you have your neutrophils that come, and the neutrophils eat things up. Right? So here you have the macrophage directly eating, for example, a bacteria, and then destroying the bacteria in special vesicles inside of it. Right? You have the bacteria is encircled in a special vesicle, which we call the phagosome. Right? And then inside the effector cell here, the macrophage has these lysosome vesicles that are filled with all kinds of proteolytic enzymes and things like that. So those two vesicles fuse so that all those nasty proteolytic enzymes and things are targeting the bacteria, and it digests it. Right? On the other hand, adaptive effectors, by and large, do not concern themselves with directly targeting and destroying the pathogen. They are more concerned with what? They are more concerned with identifying cells in our body that have already sort of succumbed to being infected. So this is particularly the case with certain types of bacteria that cause intracellular infections, or importantly, viruses, right? And in those cases, the pathogen has entered your body's cells. And once inside the cell, innate effectors like macrophages, neutrophils, or even the complement proteins that are outside can no longer get to it. So evolutionarily, you might say, well, that's just too bad then, because now this infected cell in our body, right, we're not paying attention to it. Well, yeah, but you can't afford that, right? Because those infected cells are the ones that are now going to blow up and scatter 10,000 or more new virus particles throughout your system. So our adaptive system is more focused on identifying target cells. So in this case, this cell here, okay, might still be a macrophage, right, that has gone ahead and ingested bacteria. That's why you see it in the vesicles. So this macrophage is going to present evidence of the bacteria that it found on special platforms on its surface, so that something like a T cell, in this case maybe a helper T cell, can come by, right, and take a look and recognize that this small little piece here, the little red dot, is something that its T cell receptor was designed by chance through VDJ recombination to recognize, and that alerts this T cell that this macrophage has found something, right? And this T cell then might go off and wake up other immune cells. Or this could be an infected cell, right? The cartoon might look a little different because you wouldn't expect it to have like the bacteria and little vesicles like this necessarily. But if this were an infected cell, it would also display little red dots, right, on platforms on its surface. And the purpose of that would be to attract maybe something like a killer T cell, a CD8 positive T cell to come. And then that might trigger the CD8 cell to go ahead and kill the infected cell. Okay, so that's very important, right? We've talked about B cells, we've talked about T cells. I think it's worth doing sort of a compare and contrast just to help keep the two types of small lymphocytes straight in your mind. Because both B cells and T cells are crucial for our adaptive immune response. Right? Both carry out unique antigen binding specificities, or the ability to bind unique antigen shapes based upon, these receptors that they make. But when they encounter antigen, right, and they activate, the roles that they fulfill are very different. So let's start with the B cell. Right, so the B cell that makes immunoglobulins from VDJ recombination of the immunoglobulin gene loci, it creates immunoglobulins that have unique binding surfaces on the end of them, and those B cells then might be encouraged to become plasma cells, right? So the chosen IgM that's on the surface of a brand new naive B cell. I've used that word naive B cell a couple times, but I don't know if I ever really defined that term. A naive lymphocyte, whether it's a B cell or a T cell, means that this is a brand new B cell or T cell that hasn't yet experienced anything binding to its antigen binding molecules on its surface. So a brand new B cell that comes out of your bone marrow that has immunoglobulin on its surface, right? Until that immunoglobulin has ever successfully bound to something, we say that the B cell is naive. Right? After it has bound something, depending upon the circumstances that we're learning about, then the B cell would no longer be naive, right? Because we use the words like it's chosen now, right? It's activated. So remember, the first type of immunoglobulin, or the first of the 5 subclasses of immunoglobulins on the surface of a brand new naive B cell, would be the IgM type, right? We learned that previously, that that's the first of the immunoglobulin isotypes that's made on a B cell. So the surface of this B cell would be covered with its own unique IgM, and every IgM molecule on its surface would be identical, right? Because it comes from that B cell's unique cookie recipe that it put together to make that immunoglobulin. So once it's chosen, right, it will undergo rapid clonal replication to make many copies of itself. So otherwise, this one B cell's cookie recipe is very rare, right? And now that it's determined to be very valuable, you want that B cell to make many copies of itself so you don't lose this recipe, right? Some of those B cells, the resulting identical clones, might be encouraged to become plasma cells. Right? And so really that's like the whole life mission of a B cell, is to become a plasma cell, and then it'll make the alternative RNA spliced form of its Ig that no longer is stuck to the surface. And now this soluble Ig floats around as antibodies. Here's an illustration. So here's a plasma cell. And you might be excited looking at it going like, wow, I can see all the immunoglobulins on us. No, right? It turns out that immunoglobulins, because they're small proteins, like about 150 kilodaltons, when you look at an image of a cell on this scale, you cannot see the individual small little immunoglobulins on its surface, right? So all of these little bumps are just different protrusions in the cytoplasm. But we do have microscopes, of course, that are strong enough to see it. So if you zoomed in, right, you zoom in, oh yeah, now you see individual antibodies here that are floating away from this plasma cell, right? So they're like a cloud of these antibodies coming out from the plasma cell. And each one of them you can see, sure enough, is a little Y-shaped molecule, right? So that's the function of a B cell. Ultimately, it's to get chosen to become a plasma cell so it can make antibodies. Right? That's its life's work, its life's mission. A T cell then is a bit different. T cells also start off being naive, right? They make T cell receptor through VDJ recombination, right? And most likely that shape of T cell receptor is not going to amount to much, right? Because it's just a random shape. Chances are on any given day in your body, you're not going to have a pathogen present that also presents some sort of antigen with an epitope that would be recognized by the T cell. Right? However, if you do have a T cell that's chosen, right, remember the T cell receptor never becomes a free-floating soluble form. The TCR always stays as a receptor. Right? And importantly, remember that the only way that a T cell can even use its T cell receptor to detect anything is if that thing is actually a small peptide, right? Unlike the immunoglobulins of a B cell, that are happy binding peptides or carbohydrates or lipids or any small three-dimensional shape that would fit to the end of an immunoglobulin, right? Those are all shapes that might be floating around. So an immunoglobulin that's floating around has an opportunity to bind to them. T cell receptor is different. T cell receptor is always on the surface of a T cell and can only bind short peptide, short protein fragments that are presented to the T cell receptor by another cell that is presenting these small pieces of peptide on its surface on these protein platforms, which are the MHCs. Right? And then the function of that T cell then, right, is to either kill the cell that it's contacting, right? So if it's a cytotoxic T cell and it sees something that its T cell receptor recognizes being presented by this target cell, it may send cytotoxic messages that tell this cell to undergo apoptosis, right? Or if it's a helper T cell that sees something that it likes being presented by the cell, it may then help that cell by sending other types of cytokine messages that encourage this target cell to maybe improve its function. The macrophage that was just kind of modestly eating something now gets encouraged through cytokines to really ramp up its phagocytic activity. So here you see an example of T cells, multiple T cells checking out another cell. Right? So as they're synapsing, you can almost imagine in the surface between where these two cells are touching, numerous examples of little peptides on MHC on the surface of this yellow cell being presented to the little T cell receptors that are on the surface of these pink lymphocytes, these pink T cells, and they're communicating. Okay. So this is a bit of a review over here from what we talked about last time, but this is really important. So I wanted to remind you that the T cell receptor itself, here we have a purple cytotoxic T cell, a light blue helper T cell, right? Both of them are covered with T cell receptor. So that's this light blue alpha beta T cell receptor here. Right? And that T cell receptor is designed, through the random process of VDJ recombination to be able to, by chance, detect this particular red dot, which is a small little peptide between maybe 10 to 20 amino acids or so that came from this target cell, and that target cell loaded it on top of this MHC platform, right? In this case, this is MHC type 1, right? It's made up of two components, a large kind of chain over here that goes from alpha 1, alpha 2, alpha 3, and then a separate beta microglobulin, globular protein that kind of sticks in there. Right? So the red dot is presented by this platform so that this T cell can see it. Likewise, the CD4 T cell also has a T cell receptor. And again, by chance, it may be very similar or identical in shape to the one that this CD8 cell has. So it also can bind that very same red dot that's being presented by this yellow cell up here, right? But in this case, this yellow cell is presenting that red dot on MHC class 2. That's why the MHC platform looks a little bit different, right? And we said, why would you have these two different scenarios? The one on the left is if the target cell up here was infected, right, by the pathogen that gave rise to this little red dot. So an infected cell loads little peptides on MHC1, whereas this cell over here is a professional APC, or professional antigen presenting cell, like a macrophage or a dendritic cell or even a B cell, right? Its ability to get the red dot into it, right, was not based upon the fact that it's infected by the virus or the bacteria, but maybe it got it into it because it was eating the virus or the bacteria. And therefore, the red dot, it wants to pass on this message to other cells. So it loads it on MHC2. And remember, how is it that the cytotoxic T cell versus the helper cell recognizes MHC1 or 2? It has to do, very importantly, with the fact that its surface is also covered with the co-receptor, either CD8 or CD4. And it's the right shape on the cytotoxic T cell for its CD8 to come and bind to MHC1. Right? And on the CD4, it has CD4, on the helper T cell, it has CD4 all over its surface. That CD4 is the right shape to bind MHC class II, right? And that combined signal of its T cell receptor and its co-receptor signaling then activates these T cells, right? So it's the combined signal of the TCR recognizing the specific antigen, right? In this case, the red dot peptide, and either the CD8 recognizing MHC1 or CD4 recognizing MHC2. That is what's going to activate the T cell by now stimulating the T cell to do something, right? So this slide we didn't get to last time, but it's okay because it fits in very well in our discussion today, right? So let's look at some different scenarios of what might happen here. So this is T cells that are functioning by making contact with other cells and inducing those other cells to change, right? Here we have an example of an infected cell. An infected cell, this could be any cell in your body that was infected by a virus, right? So that virus has hijacked the cell, and now this poor cell stops doing what it normally does. It's now become a factory making more virus particles, right? So that's really bad, right? Because if the cell is not stopped, it's going to release thousands and thousands of new virus particles into your tissues, right? So because it's infected, it's loading the little red dots that represent bits and pieces, fragments of protein that are coming from these virus particles on MHC1. So here comes a T cell floating by. This happens to be a CD8 T cell, right? So it has its T cell receptor on it, and it's also covered in CD8, right? Well, it's also covered in TCR. That's a good point, right? I mean, these cartoons, again, right? The cartoons oversimplify things. It draws it as though it only has one of these and one of those, but the reality is the whole surface is covered with CD8 and T cell receptors. So then when they synapse like this, right, the T cell receptor happens to be the right shape to recognize the red dot on MHC, right? And the CD8 swings over and it's the right shape to confirm that this is MHC1. Therefore, that stimulates the cytotoxic T cell to release all kinds of cytokine messages that are in the form of cytotoxic signals that tell the virus infected cell to go ahead and die. And that's how you stopped that virus cell from spreading the virus, right? Another scenario, though, you have a macrophage, or a macrophage that's busy doing its macrophage job. So it's busy eating things like this bacteria, right? So the bacteria are contained in special vesicles that then fuse with lysosomes to digest them, right? So this macrophage then is presenting the red dot that comes from the bacteria, not on MHC1, right? Because the macrophage is not in trouble here. It's not infected. It just wants to be the messenger that's passing on this message that, hey, I'm eating this thing, and look, this is bits and pieces of this thing. What do you guys want to do with this, right? So it presents the red dot on MHC2 so that if you have a CD4 helper T cell that comes by that has the T cell receptor that by chance recognizes it, and it's also covered in CD4, that combination then of T cell receptor working and CD4 swinging over and verifying that this is MHC type 2, right, this platform, that then stimulates this T cell to activate. But instead of killing the macrophage, it sends cytokine messages that do something like, wow, further activate this macrophage, right? So it causes the macrophage to just rapidly start digesting all this bacteria and maybe eat more bacteria, and importantly, send out a big cloud of cytokine messages to wake up other immune cells, right? So two different scenarios here based upon whether the cell that is presenting needs to be killed or if it needs to be helped out and encouraged to increase its function. This third one is also very important, right? Because, oh, here's our friend, the B cell, right? We talked about B cells, we talked about T cells. Let's talk about them working together. So here's a B cell that on its surface, right? This is a naive B cell that just came out of the bone marrow, and it's been gone through a process of sort of maturing that we will talk about later, and now it's going to work, right, in your body, and it has on its surface its IgM, right? An IgM that, wow, guess what? On this particular day, there's a pathogen that infects your body that has this, I don't know, this particular antigen, right? This little funny hibiscus flower looking thing. that binds to this IgM. So the IgMs, remember, are just proteins on the surface of this B cell. And just like any other protein on the surface of a cell, cells are constantly recycling them, right? They bring them in and replace them with new examples or new molecules of that protein to kind of keep things fresh on their surface. So if there's something bound to this IgM, like this red hibiscus antigen, right, it's going to get brought in with the IgM. And once it's brought in that way, right, that's not the same as the cell being infected, right, because this is actually the cell just bringing in stuff as part of recycling proteins. It will present, right, it will present the little red dots from this hibiscus on MHC2, right? So again, a helper T cell comes along, sees that it recognizes this, sees that it's MHC2, sends out these little green triangle cytokines, right? And that stimulates the B cell what? To become a plasma cell, right? Because it's telling this B cell, hey, guess what? Right? This is your lucky day. Your IgM looks like it could be really valuable. Go ahead and make the antibody form of that IgM, because up until this moment, your body had nothing, right? No antibodies that would recognize this thing. Now, at least you have these crummy little IgMs that maybe aren't great because they're just the random puzzle pieces that are close enough fit, but it's better than nothing. So this B cell starts making these antibodies as the sort of quick, first quick and dirty sort of antibody response just to get something out there and start attacking, the pathogen. Right. So we will study this interaction between T cells and B cells in much more depth, because this is one of the most important sort of events that happens in your adaptive system. And it brings together how the B cells work and the T cells work. All right. So let's elaborate a little bit more on how it is that a cell that gets, let's say a fragment of protein in it knows how to load this thing on MHC1 versus MHC2, right? Because obviously, depending upon how it ends up presenting this small little peptide, right, makes all the difference in the world as to what kind of T cell would end up getting excited about it when it sees it and causing, some big event to happen that could really influence what happens to the cell that's presenting it. Let's pretend we have a healthy macrophage. So the healthy macrophage, it has MHC1 all over its surface, just as any other cell in our body, any other nucleated cell in our body has MHC1, right? Because every cell in our body wants to be able to advertise if it's in trouble and infected, right? So healthy macrophages have MHC1 also, right? And what's that MHC1 presenting? At any given time, right, your cell is filled with all kinds of small peptides and things from your cell's own normal, perfectly normal, natural proteins that are just being recycled inside, right? And as these proteins, normal proteins, are being recycled, bits and pieces of them are what get loaded on the MHC1, right? And you might think, why would I want to do that? Because that's almost like you're trying to get your immune system to react against something that's normal. So very clearly, our immune system has to go through a selection process by which T cells that are allowed to go to work, right, are only those T cells that would not get excited about normal peptides coming from normal healthy cells being presented on MHC1. Right? After the first exam, we will talk more about things like how T cells develop, and we will learn more about this critical selection process. Right? But suffice it to say that on a healthy macrophage on its surface, right, on MHC1, it would be presenting just self-peptides, stuff that's representative of your own body and are normal and should be ignored. Because the macrophage is one of these professional APCs or antigen presenting cells, right, it is one of the few cell types in your body that also at the same time has MHC2 on its surface. Right? And that MHC2 will present not only bits of self proteins, because remember, macrophages also work as kind of custodians, right? They're going around cleaning up stuff like dead cells in your body, right? So as they take those dead cells in and recycle it, the protein pieces that come off of those are still your own body's natural proteins, right? So some of the MHC2s are going to be presenting normal peptides, but importantly, when this macrophage is eating bacteria, for example, right, and it eats the bacteria, and then the phagosome fuses with the little lysosomes, these are the little blue things around it, to form this phagolysosome that digests the bacteria. I don't know why illustrations often show it this way, like the macrophage has like a mouth end and like a butt end where it like poops out the little bits and pieces of the bacteria. But it's just meant to show you that, right, a lot of these components just get recycled and released into the space around the macrophage. But some of these small peptides will be presented on MHC2, right? So that a helper T cell that comes by gets the message that this macrophage is eating this foreign pathogen. Right? However, that's if you have a healthy macrophage. Macrophages themselves can, of course, become infected, right? So what if this is a virus that infects this macrophage? So then the MHC1 on the surface of the macrophage would be presenting the same self-peptides that they always do, that are the normal recycled peptides, but importantly, what? Now you have some of them also displaying small pieces of non-self peptides that come from the bacteria or the virus, right? So therefore, this macrophage, because it's infected, is also subjected to a cytotoxic T cell coming, recognizing the peptide in MHC1 and potentially telling this macrophage, sorry, right, you got to go because you're hijacked by the virus. You better destroy yourself. Okay. The macrophage may also be at the same time phagocytosing and eating the virus particles, in which case, the MHC2 on its surface, it would just be like presenting what the MHC2 is in the case when it was healthy, a combination of self as well as non-self peptides. But the important thing is, because it's MHC1 is presenting this peptide that cytotoxic T cells might recognize, then this macrophage itself might be told to die. So how then does the cell carefully control if it's MHC1 platform that's going to end up presenting, right, holding and presenting a peptide, or if it's MHC2? This all has to do with where the little peptides are found inside of the cell. If you think about it this way, if a pathogen was phagocytosed or eaten, then, whether it's a bacteria or virus, then its components, like its proteins, are always carefully contained in some sort of a vesicle, right? The vesicle that it originally was brought in, or even when it's digested, you have other lysosomal vesicles that fuse with the first one, and all the bits and pieces are still more or less contained inside of a vesicle, right? Compared to if the cell was infected by a pathogen, then you'd have evidence, bits and pieces of peptide just kind of floating around inside of the cytoplasm, right? Not in carefully contained vesicles. Right? So that difference provides the cell a good distinction between which peptides, either the ones that are contained in vesicles or the ones that are just floating around in the cytoplasm, should be loaded on MHC1 versus MHC2. So this cartoon illustrates that here. Let us imagine that in this bluish cytoplasm of this cell, that those virus particles, right? Those virus particles are going to be shedding proteins all over, and those proteins are just going to be freely floating around inside of the cytoplasm. Those bits and pieces of protein get transported into the ER, where they get loaded on MHC class 1. So that's how MHC1 proteins preferentially get loaded with peptides that came from just being freely floating around in the cytoplasm. For example, right, if this is due to an infection, or at any given time, more likely, it's just normal everyday proteins that are just being recycled, right? And bits and pieces go into the ER. So then they get loaded up on MHC1. The MHC1 is just kind of moved through these little vesicles, through the Golgi, and then they'll end up being stuck on the surface of the cell. On the other hand, if you have bits of peptides, like from proteins that are coming in from phagocytosis, like over here, if the cell eats something like a bacteria or a virus, then those protein products, those small peptides, are always contained right, in special vesicles. They're not freely floating around in the cytoplasm. So it's these vesicles then. In these vesicles is where MHC2 gets loaded up, right? And then once they're loaded up, the MHC2 is brought to the surface, right? So let's first start off talking about those bits and pieces of proteins that are just floating around in the cytoplasm. Remember, I said on any given moment, like right now, as you're sitting here listening to this, right, each one of your nucleated cells in your body is filled, their cytoplasm is filled with all little scraps of normal proteins, just from the normal cellular proteins that are breaking down and need to be recycled, right? Every so often, you might have one of your cells that actually has a virus that's infected it or a bacteria, in which case mixed in there in the cytoplasm would be evidence, bits and pieces of that pathogen in there as well. So those types of proteins that are floating around in the cytoplasm are recycled by the proteasome. If you have misfolded, damaged, or proteins that are no longer needed, right, they go in the recycle bin. The proteasome is the cellular recycle bin, right? It's a big, big protein complex. You can think of it as a recycle bin or a shredder. It's a large barrel-shaped complex where things get fed in through one end, and then these proteolytic enzymes that are part of the wall of the barrel go to work, snipping away at this protein that was fed in there, and then on the other end, little fragments of the protein, little peptide fragments, come out as the recycled product. In certain types of cells, especially cells that get infected, right, a different set of building blocks of the proteasome proteins are expressed, and that's what we call the immunoproteasome. So during infection, you will see an increase in the number of these special recycle bins inside there because they do a better job of trimming down the sizes of these peptides to be those short, 8 to 15 amino acids or so that fit nicely on MHC1. So here's an image of what a proteasome really looks like. So I think this is really cool, right? So you can really tell the central part is the main barrel part, and then these big elaborate purple and navy blue ends are these caps. And literally, these caps can open up. Proteins, misfolded proteins can be fed in, and then these central enzymatic units here go to work, snipping away, chewing up the protein into small pieces, and those little pieces are then released. Okay. So let's look at how these little pieces then make it from the proteasome into the ER and how they're loaded up. So we said in the endoplasmic reticulum is where MHC1 gets loaded. Right? So MHC1, it's just a protein complex itself. And like many proteins in our cells, right, it finishes being put together in the endoplasmic reticulum before it's moved out somewhere else to do its job. So here inside the ER, so the white space is the lumen of the ER. So inside the ER membrane compartment, you have MHC1 that's being assembled here. So remember, MHC1 consists of one long chain here. I know it's kind of funny the way they kind of draw it, but just think of it as one polypeptide chain that has these 3 different shaped globular domains in it, right? So to remind us that the first 2 domains actually make the cleft that hold the peptide, they draw it this way, right? And then the 3rd domain is more of like the stem part. Right? There are certain proteins in the ER that help put this thing together. Proteins like calnexin, calreticulin, ERP57. You don't have to know the names of those, okay? But I want you to get the impression, though, that the MHC1 is being put together in the ER, and then here's that separate little globular beta microglobulin group that's going to come and together, along with that main chain, form the MHC1. All right. Here out in the cytoplasm, you have one of those proteasome barrels, recycle bins, right? And here you see being fed into it is a scrap, big, long scrap of some protein that's all misfolded. It could be a normal protein that just is being recycled, which is happening right now, right, in your body. Or it could be a protein that comes from a piece of virus or bacteria that's floating around in the cytoplasm because the cell is infected by it. The proteasome chews it up into small little peptide fragments. Right? These peptide fragments then go through this special little gateway. This is what we call the transporter associated with antigen processing gateway, or TAP. So that's how these peptides come in. And there's another protein called an ER aminopeptidase, which edits. So this is an ERAP protein. It edits or trims these little peptides down to be even smaller, so that they might have started off as like 30, 40 amino acids long. That's too big to load on an MHC platform. So proteins like the ERAP enzyme cut them down, so they're like nice 8 to 15 amino acids. And so that's why they become the little red dot, right? And the little red dot gets loaded on the MHC1. And then the MHC1 that is now loaded with the red dot, right, it kind of buds off as part of this vesicle that then travels to the cell membrane. And once the membrane of the vesicle fuses with the membrane up here, right, this MHC1 is now on the surface of the cell, presenting the red dot that was loaded on it in the ER, right? So this way, right, you have only the peptide pieces that started off floating around in the cytoplasm. These are the things that are presented on MHC1. Here's another cartoon that just illustrates the exact same thing. I like to sometimes show you guys different ways of visually presenting things, you know, because cartoons can get you into a lot of trouble, right? If you only see one cartoon and think this is what it looks like, right? No, this is the same thing, but just presented artistically in a different way, right? So here's the MHC1, the main chain of it here in orange, then little beta microglobulin together makes the completed MHC1. You have the proteasome making big peptide fragments from a huge protein that was unfolded and fed through it. Look, you can see the caps. The caps are open of the barrel, right? Then these fragments go through the TAP little gateway. They're worked on by the ERAP to trim them down to be the small, 8 to 15 amino acids or so, loaded onto the MHC1. And then the MHC1 buds off as a vesicle, goes through the Golgi network to be properly labeled for transport to the cell membrane, and eventually it ends up on the cell membrane, where we happen to have a cytotoxic T cell that comes by and it recognizes it. Right. So what about MHC2 loading? So MHC2 loading happens to proteins that are taken in, right, by the cell, like through phagocytosis. So here you can see this protein was taken in. It always stays in a nice vesicle, right? It's not floating around in the gray cytoplasm. So when it gets lysed and chopped down by, let's say, the lysosomal enzymes, it's here that, look, another vesicle comes cruising up to it. And this vesicle has MHC2 in it, right? So in MHC2, its vesicle fuses with the phagolysosome. That's when these red dots get loaded on MHC2. And then this MHC2 will continue on to being presented on the surface, right? So importantly, MHC2 is also assembled in the ER. I mean, it is just a protein, right? But when it's in the ER and is fully made, it's plugged up by this green protein here, which is known as the invariant chain. And that prevents it from being loaded up by other fragments of peptides that are also in the ER, right? Because the MHC1s that are in here are being loaded up by peptides in here. But the MHC2, you don't want it to load up with peptides yet, right? As a vesicle, it floats away. There's a little piece of that invariant chain that stays plugged in it like a cork on a wine bottle, right? Once the phagocytosed contents of this endosomal vesicle fuses with it, right, and you have all these little red dots, then the cork is removed and these little red dots can get loaded on it before it's transported out. So again, this is just another way to contrast the two. All right, so oops, sorry. So we're kind of out of time. So this is just letting you see, when you have time, take a look at this, because this is, again, the loading of MHC class 1, right, where it starts off in the ER and where the little, in this case, little beige dots that represent peptide that came from a virus or some other bad evidence of disease was processed by the proteasome, went through the ER where it's loaded up on the MHC1, and then when it's presented on the surface, it's a reminder that that's what cytotoxic CD8 T cells will recognize, right? On this case, on the right, MHC2, even though it's made in the ER also, right, it's plugged up, so it doesn't get improperly loaded up yet, right? It's only after it has a chance in this vesicle here to come in contact with the same virus or other disease evidence antigen, but in this case, this was taken in through the process of phagocytosis, and so it gets loaded up on MHC2. so that now when it's presented, you favor the recruitment of CD4 helper T cells to come and recognize it. Okay? All right. I'll save the next slide for next time, but we'll go ahead and stop there today.